And we can now start the presentation. Hello and welcome everybody to our webinar, the first one of the Permeability series this year, with the title The Concept of Permeability in Drug Discovery and Formulation, a concept often misunderstood. My name is Ali. I'm a Sales and Distribution Manager here at Inome and Fabioc, and I will be the host for this webinar and all the following ones. And on today's agenda, we will take a closer look on topics that come again and again in conversations and meetings with customers, from the basic questions on what permeability is to analysis and interpretation of the apparent permeability and other numbers, facts, and figures we get. We are very happy again to have Professor Massimiliano Piodicagno as our presenter today for this live webinar. Max has a very impressive academic vita and can therefore be called a total expert in pharmaceutics and especially in drug formulation and permeability. Next to that, Max is also a freelancing scientific consultant for our company Inome in Germany, the company which manufactures and commercializes PermeaPad. Max did co-invent this product together with Professor Annette Bauer-Brandl back at the Southern Denmark University, where he used to work. And as you can see, I did not overstate by calling Max a total expert. So before we start right away with the webinar you all been waiting for, a few things regarding the organization. You can type in any question in the Q&A section or in the chat, and we will try to answer all of them after the presentation. So without further ado, Max, the word is yours, and to all other people, have fun with the presentation and see you later again.
Yes, thanks a lot, Ali, for this nice introduction. I hope that you all can hear me well. Is it the audio good, Ali?
Yes, it's good.
Okay, so today I would just, my intent is to try to give an overview mostly on the concept of permeability and apparent permeability, that are the two, or that is the parameter actually that is most used in order to somehow predict in vivo performance of chemical entities, as well as enabling formulation when permeability studies are utilized. So I will start with defining the concept of apparent permeability, give some example on which types of permeability do we have, which are the barriers that actually can be employed for measuring such parameter, relevant for pharmaceutics, of course. And I will go through the general misconceptions on permeability. We can go to the next one, Ali. So, in general, in the past, it was common practice to utilize the octanol water partition coefficient or distribution coefficient, if this was measured for electrolyte or ionizable compounds. But as probably all of you know, this parameter is definitely not a good parameter for predicting the in vitro performance of a drug. So, for example, this is not enough to predict bioavailability. So, in the last years, another concept has arisen, and this is called permeability. The permeability is measured through biomimeric barrier, some of cellular nature, and some that are actually biomimeric. PermeaPad is one example of a biomimeric synthetic or artificial barrier. You can go to the next one, Ali, text. And here you can see the permeability that refers to both cellular culture systems and artificial barriers such as PermeaPad and Pampa, but I will discuss it later during the presentation. You can jump to the next. So, what is permeability? Can we give a definition to permeability? Permeability comes from the Latin word or verb permeare, and this means to go through something. In general, the permeability, if we search for scientific literature, permeability is often much more related to material science and magnetic field, so physics, more than pharmaceutics. But in the latest years, let's say in the last 20 to 30 years, this parameter has become very, very much implemented also in pharmaceutical technology. There is, as it is not a thermodynamic parameter, it is kind of difficult to define. So the highlight in red is my personal definition of permeability. So it's a conventional parameter that is able to describe quite well the ability of APIs and chemical entities to diffuse or transport through a biomimetic barrier. And this parameter should actually correlate quite well to in vivo drug performances. So it's a predictive parameter. And as you can see in the slides, here is it's just a search I made on Scopus utilizing the terms permeability, and you see that before the 1995, the number of citation or the number of reports publication with permeability were very, very low, around 1,000 per year. But then, Ali, if you click 3 times, we got 3 major achievements in the pharmaceutical research. First of all, Amidon introduced the BCS classification system in 95. Then Kansi et al. Introduced in 98 the PAMPA, the parallel artificial membrane permeability assay. It is the first official biomimetic barrier of not cellular nature. And then in 2001, Lipinski introduced the famous rule of 5. And you see that after the 2000, basically the permeability in terms of publications has really exponentially grown. Can you go further again? Okay, so when we talk about permeability, when this terms was actually introduced for pharmaceutical purposes, but I think that the first official introduction was made by Amidon et al. In the same article where they actually introduced the biopharmaceutics classification systems, and it was defined as the human permeability. So in this work, in this very elegant, brilliant piece of scientific work, the flux, the gastrointestinal flux was correlated to a parameter called human permeability and the concentration of the drug at the membrane, so at the intestinal wall surface. And if you click further, Ali, they actually forecast that such a parameter, if this parameter could be possible to be measured, so the permeability, the human permeability should actually correlate very well with the fraction absorbed. Of course, it's a parameter that is this human permeability is a sort of ideal parameter because it is not possible to measure unless we actually extract that from the fraction absorbed. So, but actually the apparent permeability is a concept that is built on this. And if Ali, you go further, the next slide, this led us to the description of how a permeability experiment is generally run. Permeability experiments are run utilizing diffusion cells. So cells, glass in quartz, in glass, in plastic, that actually are generally mostly all built with the same structure. So we have a donor compartment that can be stirred or not. Then we have a barrier, a biomimetic barrier in the middle, the yellow region of the figure, and then we have an acceptor compartment. What actually is, if we place a solution of a drug in the donor compartment, the drug molecules will spontaneously move. You can click, Ali. We spontaneously move through the barrier, following a diffusion that can be somehow approximated to be a Fickian diffusion. So it could approximately be described by the Fick's first law diffusion, where the flux of the drug molecules will be basically the diffusivity constant of the drug in the barrier multiplied by the concentration gradient. Now, if we actually are able to measure this flux, then if we divide this flux by the initial concentration of the drug, we obtain the so-called apparent permeability coefficient. Can you go further, Ali? So again, so in theory, what in practical, what we do experimentally is that we actually measure with different analytical tools, HPLC, UV, whatsoever, the concentration of the drug that accumulates in the receptor. And we convert this in a quantity, millimole, micromole, grams, and we actually build a diagrams like the one you saw on the right, top right. So amount accumulated of surface area over time. This is a classical profile where we can see that we have actually a latency time. I'll if you click a steady state and a non-sync condition, because generally the top, if the experiment is too long, then we lose the sync condition and we lose the linearity. But if we can get this steady state region where we have a good linearity, then here the slope of this curve will represent your flux. And this can just be normalized, Ali, if you click over the concentration and get the upper permeability coefficient. The figure below represents basically the flux at any time point, and you can see with this nice representation that we can actually we should measure the permeability in this way when the flux is constant or approximately constant. Can you go ahead, Ali? Of course, I said before that to measure it, the measurement of the permeability is based on the physics of diffusion. So we assume that the Fick's law of diffusion is actually functioning here. Now, this is a serious approximation. First of all, we have at least 3 different phases, because the molecules move from a water phase to a lipoidal phase, the barrier, again to a water phase, crossing at least 2 interfaces. So in theory, this elegant equation should not be applied. But we see that experimentally it can hold. It can hold if we make some assumption. The first is that the concentration gradient need to be constant. So the concentration during the experiment and the difference of concentration within the receptor and the donor and the thickness of the barrier need to be constant. The second approximation is the concentration of the donor should be much larger than the concentration of the receptor. Then we can even more simplify the equation, because the flux in this case become dependent on this average diffusivity multiplied by the concentration of the drug in the donor divided by the thickness. The third approximation is the one I named before. D is constant. Of course, this is not true. We have at least 2 different diffusivities, but in the experimental setup, we never take into calculation this parameter diffusivity. And then, if we assume we accept that this parameter is constant, then we can calculate the permeability with the equation I showed before, and I report here again, divided by the flux over the concentration of the drug in the donor. Please, Ali. So, in theory, actually, the system of diffusion is much more complex, and this is a kind of better representation of what is happening. We have the concentration of our drug in the donor, but then we have an austere water layer that applies on the barrier, and also on the other side of the receptor. Of course, if we have a steering, the austere water layer of the drug in the receptor compartment will be lower than the receptor, than the austere water layer in the donor compartment. But anyway, it should be taken into account. And this actually drive us to the concept of effective permeability. So the effective permeability is actually, we cannot actually sum together the permeability in the different layers. But if we consider the permeability barrier as a series of layers, 2 unsteered layers in this case, and 1 diffusion layer, then we can actually utilize the resistance, so the opposite of the permeability. Ali, you can click. And then we can actually find the so-called effective permeability, that is nothing more than the permeability that is measured inside the barrier itself. And this is of importance because it's extremely important. Of importance is to understand, because there are drugs that are more limited by the permeation by the barrier, the lipoidal barrier, but also drugs that are limited by their diffusion in the austere water layer. Can you go? You can go further, Ali. So again, if we zoom into the effective permeability, as I said before, when we talk about the semi-synthetic barrier, but also the cellular-based system, um, this is a simple representation of how a barrier is generally built. We have a support layer, a support, for example, in filter in nitrocellulose. Then we have a lipoidal layer that in CACO2 cells is represented by a monolayer of cells. And then we can have an additive support layer on top. This could actually be, for example, just the mucus, or can be an additive layer of protective that protected the barrier itself, like in the case of PermePET. Can go ahead. And here is the representation of the real effective permeability, because if we have different supports or different layers, then really our effective permeability should be represented just by the lipoidal layer of the barrier. So which are the biological barriers that should be most relevant to mimic with such says? Well, if we again utilize Scopus and make a search, we see that definitely skin and gastrointestinal barrier are the most investigated one in terms of permeability and bioavailability, but also oral mucosa and nasal mucosa are also now raising importance. When we actually come to the commercially available barrier, we need to eventually distinguish between biological and artificial. The biological, I reported just for simplicity, the CACO2 cells, so the colon carcinoma cells model, introduced by Arthurson back in 1990, early 90s. But of course, nowadays we have a lot of other type of cells that can be grown on a monolayer can give us a value for permeation. Episkin is similar concept of CACO2 cells, but it's mimicking the epidermis. Then we can utilize ex vivo tissue, so tissue that comes from animals. This is of course not alive, even from humans eventually. One of the most implemented is in skin studies, and is to utilize the ear of the pigs, and this is commercially available. When it goes to artificial, then we have the so-called PAMPA model, the first one that was introduced, the PAMPA barrier, the parallel artificial membrane permeability assay. And then we have the newly introduced PermePED that has a different design, and I will come back to that later. We also have to mention two membranes. One is the regenerated cellulose membrane, that is also used somehow to study permeation, but this should be clear that in general these type of barriers are not biomimeric barriers. They are more like a size exclusion barrier, ranking drugs according to the size, and the transem, that is similar concept used for skin studies. And this is just a nomenclature difference between what is a membrane and a barrier. So if you go ahead, still click, you can go ahead. So back 1 second. So when we talk about membrane, we indicate a thin sheet of tissue that is acting as a boundary, like a monolayer of cell is a membrane. But this monolayer of cell is built on a support layer of nitrocellulose. Oh, this is the barrier. Okay, you can go ahead. And when I say that actually the barrier like regenerated cellulose, dialysis barrier are not really biomimeric, I show you an example here. These are the permeability, upper permeability measurement that has been performed of a series of drugs on two types of regenerated cellulose with different cutoff, 2 kilodalton and 15 to 20 kilodalton. As you can see, the permeability is reduced over the molecular weight in a sort of an equation 1 over x. The slope of this changing of the curvature is different, and this is of course related to the size of the pore of the cellulose, but in any case, this ranking is not really a biomimeric ranking. This is just telling us that the larger is the molecule, the lower will be the permeability assessment utilizing this type of barriers. And another example, if you click Ali, comes from experiment we have run within utilizing PermeaPad and the size exclusion barrier, the PermeaPlane that also Fabio commercialized for ketoprofen. So ketoprofen is an acidic drug. It has a pKa around 4.4. And it's rather lipophilic. So if we measure the permeability of ketoprofen through a biomimetic barrier, we should observe a different pH. The permeability should be slightly higher at pH 2 than at pH 7.4, because this is an acidic drug, and in theory, the unionized form of the drug is more permeable. And in fact, utilizing PermeaPad, we actually see this difference. It's not a large difference, and the reason of that is that this experiment were actually run with a special setup. I will show you later. But in any case, ketoprofen is considered to be a medium permeable compound with a slightly better permeation in vivo in acidic environments. If we utilize regenerated cellulose, we don't observe this, because the regenerated cellulose is will just give you a higher permeation if you generate a higher gradient of concentration, like you see in the other slide, where using regenerated cellulose we measure a higher permeation for in pH 7.4, and this is an artifact. This is not actually what should be in real condition. And as I said, this experiment I showed before were measured with a new diffusion, a new flow through diffusion cell system. It is called the MIVO. The MIVO, I report here a figure. It's basically a cell where you actually have a donor compartment where you actually place a solution. In the middle, you have a barrier. It was designed for utilizing biological cells, but we adapted this for utilization of PermeaPad. And in the bottom, you have a receiving chamber, but this receiving chamber is a flow through cells. So basically, what we do here is that we eliminate the unsterile water layer in the bottom, in the receptor phase, because we always have a continuous flush of the environment, and that's why we get a lower difference permeation, because ketoprofen in its ionized form is affected also in its total upper permeability by the diffusion in the unsterile water layer. Removing the sterile water layer reduce the absolute value of permeation. And there are different ways that to account for the unsterile water layer that has been actually done before. Already in this work from 2007, of Broster et al., we see that different drugs applying a different steering. If we apply a different steering, we generate a different steering water layer thickness in the acceptor media, and we get a different value of effective permeability of the drugs. So this is not surprising because, of course, as I said, all these assays that are in multiple plates or diffusion cells has the limitation to be somehow I lost the presentation, Ali.
Sorry, my internet just broke. I'm just sharing my screen.
Yeah.
Sorry.
Okay. Yeah, what I wanted to say is just that, as you see, creating a different sterile water layer, we create a lower permeation. And this is, of course, related to the drug. For example, hydrocortisone has a less, and carbamazepine are less influenced, but anyway, having a thicker unsterile water layer reduce the permeation. And if you click Unique, you see that another work, this is of after a little, also from 2004, no, back one slide. No.
Forward
Forward one slide, this one. This was also an elegant studies where actually they have tested the permeability of drugs, of different drugs at different steering rate, and they also were able to come out with these equations where they correlate basically the steering efficiency factor and the steering speed with the actual permeability you measure in such permeability assays, especially for multiple plates where you have very small volume and high difficulties in steering. Yes. And this last point I want to make is the measurement itself of the permeability and which concentration should we use to measure the permeability. Ali? Yes. So this is a study that has been performed not on biomimetic barrier on biomimetic barrier, but ex vivo. And actually, this is just a slide that highlight the fact that when we talk about permeability, it's very important to agree on the way we calculate it. So in this case, hydrocortisone was formulated in 3 different types of enabling formulation: liposomal, micellars, and polyvinyl pyrrolidone solution. And you see the profile of permeation are very different. So if you click on it, we actually measure the concentrations and the permeability, and we saw that the best fitting of the flux was not with the total amount of drug that was dissolved, but actually with the molecular dissolved fraction of hydrocortisone. So my advice is, in general, to always try to account for the concentration of the different species we have, and in this case the free molecular fraction, and utilize this to measure the permeability, because the permeability in theory it is not a thermodynamic parameter, but in similar condition it should reflect the interaction of the drug and the barrier. So it should be a kind of a constant. If we utilize enabling formulation, this value will be much different because the upper permeability can be enhanced of folder of magnitudes, but this will not reflect the real upper permeability. Yeah, so concluding this short presentation, I would say that upper permeability can be considered as a qualitative parameter, but also quantitative if we can take into account all parameters of the permeation. In vitro non-cellular models are definitely or can provide useful information, not only for API development, but also in pre-formulation stage for enabling formulation. But it is always important to know the system and take in account all the variabilities, for example, the unstable water layers. And when enabling formulation are investigated, consider to apply non-linear data fitting approach is also a possibility, because especially with enabling formulation, we are not expecting a steady state flux, but we are expecting a non-linear behavior. And with this, I would like to thank you all for the attention, and I will be happy to answer to questions if there are any.
Also from my side, thanks, Max,